Okay, very good afternoon to all of you. And it's a very, very long session, so I can see that your faces. So again, by the way, I won't be taking much time because just before me, you heard Professor Dua. So Professor Dua and I actually collaborate extensively. So much of what I'm going to share here is also what actually is achievement of Professor Dua. Okay, so having said that, I'm not going to take much of your time. Okay, so what I'm going to present here is that what actually as a researcher, what we did, especially on this topic, over the past 1 decade. So over the past one decade, we worked heavily on, say, nano-based research, nano-based innovations actually. So, and my area actually is inflammatory conditions, and like, obviously we know that inflammatory conditions like asthma or lung cancer, fibrosis, all these are inflammatory conditions. So that's my area actually. So I'm not going to present about everything here. I'm just going to take one part of it, which is lung cancer. So I actually come from this particular university, which is called as International Medical University. Okay, so this university is located in Kuala Lumpur in Malaysia. So I've been working here for the past 15 over years. And I also want to highlight my research collaboration team. So it's only because of the team I'm where I am today. Okay, so like what Professor Dua mentioned earlier, it's very, very important to collaborate. So this is my team. So my team actually, actually is spread across the entire, it depends on what kind of work I'm collaborating with. So in terms of lung diseases or respiratory inflammatory condition, this is my team. So obviously I have whatever the work, what you probably will see on the internet, or say for example Scopus or say Clarivate. Whatever you see actually is all because of the collaboration with this particular team. So Professor Dua already have highlighted about a few of the achievements of the team. So why I'm actually telling or emphasizing here about collaboration is that you see that I'm actually in this field for the past maybe 15 years or so, okay, or say especially in this field of say intensive research for the past 10 years. But these 10 years have taken me to really greater heights because of collaboration. If it were not for the collaboration, I wouldn't have reached this level actually. Like how they introduced me just now actually, so my H index is actually 73. Citations are more than 23,000. So how did they come? How this came because of collaboration. So my sincere advice will be to anyone here doing research, try to collaborate if you're not yet collaborating. So collaborate with people, collaborate with national, international people, so which will really, you can see the outcome in whenever you actually, the impact actually, you can see very soon when you start to collaborate. So if you don't collaborate, you are the loser actually. So that's my sincere advice here. So as I mentioned to you, this will be my area of research predominantly. So I generally work on inflammatory conditions and specifically on respiratory inflammatory conditions like asthma. I worked on asthma, lung cancer, and we also work on COPD. Okay, so this is something which now I'm currently or heavily involved in. Okay, especially on inflammatory conditions of lung or say lung diseases. So today I'm just going to take one part of it actually. The work which I did is on lung cancer. So I'm sure many of us know what exactly is lung cancer. So lung cancer actually is one of the most deadliest forms of cancer. In terms of mortality, it actually ranks number 2. Sometimes some sources say it's number 1, but many times we see that lung cancer actually ranks number 2, especially in terms of its mortality rate. People dying because of lung cancer. And if you think carefully, and a lot of these deaths come because of one main problem, which is smoking, and secondly because of industrial pollution. And this very much applies to a country like India as well, because a lot of because of the pollution actually. I'll show you some kind of statistics where it shows why I mentioned the name India actually, because Asia here, if you see the statistics here, actually has a very high percentage of existing cancer cases and also newly identified cancer cases. So you know that this actually is a very serious issue, especially in countries which are in the Asian region actually. So lung cancer is one of the most deadliest diseases actually, which is actually affecting this area here, this region here. Some of us might have already known much about lung cancer actually, so these are the factors that increase the cancer risk or the risk of getting lung cancer actually. And if you see the number one culprit here is air pollution. Pollution, this morning we were driving from the hotel to here. Yeah, I could see the amount of pollution and it's literally maybe less than 10% visibility. So I was thinking it's actually mist. You know that we see that the romantic movies, mist kind of thing, but look, the driving said it's not mist, it's actually smog. Smoke combined with fog, it's called smog. So actually it's not mist, it's actually kind of a polluted atmosphere. So actually it's very, very concerning because the number one culprit for the incidence of lung cancer is actually air pollution, the quality of air we breathe. That is the first one. Second is actually you have a lot of this, we sit down here, radon, diesel exhaust, asbestos, other chemicals, genetics. Second-hand smoke also is one of the main factors increase lung cancer risk. These are the 4 stages of lung cancer. I guess some of us are familiar with this. Stage 1, stage 2, and stage 4. And we see that as it progresses actually, and say after stage 2, the survival rate kind of drops significantly. Just briefly giving you the information about lung cancer. So this is the pathophysiology of lung cancer. So there are several markers, several pro-inflammatory cytokines which are involved, especially in the progression of disease or causing or pathology, especially involved the pathology of the pathogenesis of lung cancer. So you have several proteins which are involved, several genes which are involved, linked to actually which can lead to lung cancer. So these are the main types of lung cancer. Around 80 to 85% of lung cancers are actually the small cell lung cancer. We call it NSCLC. It actually contributes to 80 to 85%. The remaining 10 to 15% actually is contributed by the small cell lung cancer. So the non-small cell lung cancer is the one which actually we have further subtypes. We have adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. All this fall under that major type of non-small cell lung cancer. The other one actually is small cell lung cancer, which contributes to up to 15%. But the problem is the small cell lung cancer, it grows and spreads faster than the non-small cell lung cancer. So this is the current existing therapy what we have, the interventions what we have. Majorly, they are actually treated by surgery, chemotherapy, sometimes immunotherapy using hormones, and sometimes even targeted therapy. But the problem is that a lot of these interventions or treatments, they have their own side effects. They have their own adverse reactions, and many times these side effects actually are not. People don't recover from the side effects. That's one of the major problems with the current existing therapy. So that's why we need more people like you, to become researchers who can actually work in this area to kind of extend science so that we have proper therapies for these cancers, especially lung cancer. One area which we can see that is natural sources, or say, for example, from nature, for example, plant. Another one is marine. because these are actually places where it's completely not yet tapped well. We only hardly can find 10 to 15 percentage of plant sources which have been tested for this kind of diseases. And similarly, if you're talking about marine sources, less than 5% of marine, you know, biomolecules or say organisms are being tested or have been tested for such kind of problems. So we have potential, a vast potential to kind of translate research. But we need to take the initiative to do the research. So over the past 10 years, so these are some of the natural phyto-based drugs, phyto-based constituents which have been working with. So starting with say beta-glycerinic acid, this is one of the very potent natural products available from or is present in this particular plant called licorice. I think most of us know this licorice plant. And just now Professor Doe was talking about berberine, from berberis. This was another active molecule which I worked with, and then we also have cerumbone, which recently we worked. It's from Acorus calamus plant. So all these are actually plant constituents which have shown a vast potential. They are very promising potential. They have shown promising potential. So that's why I'm telling you that we have, especially a country like India, we have so many types of plant families, especially which we can't find other parts of the world. Because India is one of the certain parts of India actually is one of the recognized biodiversity spots where we can find plants which we can't find elsewhere. So we have a large pool of plants to work with. So these are some of the plants. I'm not going to all the plants which we have been doing. Obviously, we also worked in turmeric, curcumin. So we also worked in some other plants as well. But these are some of the most recent findings what I actually are showing you. We also worked in this particular tree bark called as agarwood. So we had taken the oil of this particular tree, agarwood oil, which also has shown potent activity. And this was the one which we had actually gone for PCT. We had patented this actually. So these are some of the plants which are natural molecules or biomolecules which we had worked in the past 10 years, which had shown potential for lung cancer. So this is a study which actually we published in the journal JBDST. So this is about the 18-beta-glycerinic acid from licorice. So we have actually conducted. So what generally we do is that like what I told you, I collaborate a lot. So our lab in Malaysia, what we do is we actually prepare or formulate nanoformulations. And then once we have the nanoformulations, we send it over to Professor Dua's lab. So Professor Dua will actually conduct the biological study. So that's how we collaborate. See, it's very easy to collaborate that way, right? So wherever or whichever area you are strong, you do your part and then you can invite another lab to collaborate. So what we do is we actually prepare nanoformulations. We prepare different types. Now we have a whole range of nanoparticles and nanoformulations. Okay? So we actually prepare nanoformulations. So this one also was done in a very similar way, but here we use PLGA nanoparticles to find out if the combination or when this glycerinic acid is loaded with this PLGA, if it can actually produce some kind of anti-cancer, especially in lung cancer. So we use cell line here, A549, if you're familiar with that, that's actually a lung cancer cell line, A549. So we use A54 cell line to check whether this molecule, this biomolecule is effective. It's actually very effective. You wouldn't believe the concentration of this molecule which was effective. It was 5 nanomoles. 5 nanomoles of this 18-beta-glycerinic acid actually showed amazing results. So this is how actually the mechanism of this particular biomolecule works. Actually it has inhibitory activities on several genes which are actually oncogenes, what you see on the left top. is oncogenes. So it actually inhibits these oncogenes. At the same time, it actually upregulates this gene MLKL, and also some of the proteins which are actually contributory or pro-inflammatory also, this particular molecule will inhibit. So that's what the entire outcome is. It actually produces a very good, significant anti-inflammatory effect, which is anti-cancer effect. So another molecule which we worked was on zerumbone. Zerumbone is found in many of the tuberous plants actually. So this one actually from Acorus calamus. So we found actually very good activity here as well. So here we use a model which is liquid crystalline nanoparticles. So we actually found out that they actually inhibits the proliferation and migration of non-small cell lung cancer. So this is a mechanism again here. So we found all these findings, especially on this zerumbone nanoparticles. And majorly, most of the studies involving this, you can see that the pro-oncogenes also, they will be down-regulated. Okay, so we see that all these pro-oncogenes or pro-inflammatory particles are down-regulated, eventually producing that kind of anti-cancer effect. So not only that, we also have this one which we studied on berberine, again another national molecule. So we saw that, or we found out that actually berberine actually downregulates all these components here, because all the osteopontin, and then the estrogen receptor, all these actually they are actually carcinogen, or they cause aggravation of cancer. So you can see here that it can be used in this study A5 cells. So it actually, first of all, it actually reduces the proliferation, especially of using through this carbonic anhydrase, reduces the cell survival using the cancer cell survival, and also it actually downregulates metastasis. So final outcome, overall outcome is that berberine, liquid crystalline nanoparticles, actually helps with anti-cancer potential. And this was another model, actually, cytotoxic mechanism of berberine phytotriol. That's another carrier which we use. So even in this one, actually, it produced significant results. So natural products actually have plenty of potential, especially, okay, which is still untapped. This is another paper which we published. Berberine nanostructures attenuate beta-catenin, which is a key component of epithelial mesenchymal transition in lung cancer, which is lung adenocarcinoma. So, all in a nutshell, actually, all these papers actually, we have found out that nature actually has this kind of potential that sometimes even the normal or commonly found synthetic drugs may not have. And also one more thing is that synthetic drugs also may have, other effects. Whereas nature drugs from nature, I don't say that drugs from the plants are safe, completely safe. I don't agree with that, because we have plants from the nature also which are extremely toxic and poisonous. But there are some plants which can be safely, we can actually administer once through proper clinical research. That's much of my study in the past 10 years. And this is, we have been, our team actually have been constantly, throughout the past few years and if I properly remember, our team actually has been in the top 2% for the more than six years. And this is one study which actually I would like to highlight, which was recently published in the journal Molecular Oncology, where our study was actually portrayed on the cover page of the journal. So I was talking about collaboration. So this is our the map of how we collaborate. And you see, this is actually not published by us. This paper is not prepared by us. Somebody else prepared these papers. And what these papers are talking about, I don't think you can see very clearly what these papers are talking about, whom we are collaborating with, and what is the impact we receive. So if you see on the left-hand side, so this actually is, it says the global research and current trends in nanotherapy in lung cancer research. So it's not actually we didn't submit this paper. And once it was an accident, we bumped into this paper. We saw that Dr. Dua is there, and my name is there. So it says that these are the people who are actually working predominantly in the area of lung research. Similarly, on the right-hand side, the paper, what you see, we saw a cluster. Our entire team is there on the paper. And we can see Professor Dua, my name is there. I mean, all these are actually the papers. We actually work with very closely for the past almost 10 years. So this is our team in Australia actually who did predominantly on lung research. So the paper which I showed a while ago on licorice research that actually is done by the person who is in the picture on the right, left to Dr. Dua. So he's not a student who actually did the research. So all these are actually people who actually collaborate. So they do work in my lab, and also then they go over to Professor Dua's lab and also do their work there. So here again, this is one of my students, and this girl actually is from Sri Lanka. So she actually did her master's under me, and again collaborated with Professor Dua's lab. So this is where actually Professor Dua is working now. And similarly, with Queensland University of Technology, we have some studies done there as well. All this actually will emphasize, what I'm emphasizing, it all comes because of collaboration. It's only because of collaboration you see that we can work extensively, and it's actually impact. This is what I mean by impact. So when we have impact, so when there is impact, you get more invitations for collaborations. So I really want to urge you, keep that word, collaboration, in your mind. It is very, very, very essential, especially if you're a researcher, if you are an undergrad student, postgrad student, who maybe doesn't matter. Collaboration is very, very essential. Otherwise, you can imagine, I've been doing research for 10 years, but within 10 years, I reached these heights, especially in terms of number of publications or citations. It's impossible without collaboration. Yeah, so please keep that in mind. I think with that, I will want to conclude my talk. It's been too late. Thank you so much for your patient listening. Thank you.
Thank you so much, Dr. Dinesh, for sharing your insightful presentation. Now I would like to request Dr. Raghu Pratap Singh and Dr. Jafar Khan to kindly felicitate Dr. Dinesh for the wonderful presence.
Very good question. Thank you for the question. So basically, we choose a drug which already has some kind of activity, and what I do is I look for anti-inflammatory activity. So curcumin was identified that way. So I checked the literature. Already there were several papers on curcumin, which already was published on anti-inflammatory activity, not more than that. Anti-inflammatory activity. Then sometimes we also might end up with some publications where they have conducted, say, anti-inflammatory activities or say related activities like asthma or activities which they have shown that it is actually very potential. But then you see the methodology, they have taken raw curcumin or they used some other kind of curcumin. So my idea was, why not try with a nanotechnology to enhance the properties. So most of these drugs, one biggest problem is that solubility. Solubility is a very important, especially curcumin, berberine, all these are very wonderful miracle drugs. But problem is solubility. Solubility in the biological fluids is extremely low. So we need some kind of innovation to enhance the solubility. there comes the role of nanotechnology. So that's how we choose the molecules, or we choose the drug in that way. So, and then when we find out and when we compare with already existing studies, and we see drastic difference, especially in absorption, bioavailability, when all these, the parameters are very high, especially when you convert it into nanotechnology. So this is how basically we choose. We don't choose any other way. We first search for papers for a specific drug. If it has anti-inflammatory potential, that is very important. Only then we move on. It's impossible. We cannot control that. What we do is what we can. So we only do on, say, certain inflammatory pathways, because given the time duration, given the supplies, given the funds what we have, we only can do that much. But we can further progress continuously to do a large scale study. But we cannot do that. But what we do is, okay, we can do at least this much for the progression of science.
We are also working in the lung cancer research opportunity. So whenever I am something like because of what solution they are saying that have you performed initial study design or initial design, then they are asking why you not perform kinetic. So it is never ending. But whenever you want to do the science application, because the publication is not the end. On the basis of publication, we can carry out the research for their translation. Right? But science is because thank you so much for sir for giving wonderful explanation of the
Light on argan oil. Just light on argan oil. How argan oil. How do you
Forward for the kind felicitation.
Thank you so much. So our final speaker for this plenary session brings attention to the natural product research in the context of neurodegeneration and aging. We are pleased to invite Professor Dr. Devin Tenkom Now, Dean of the Faculty of Allied Health Sciences at Chulalongkorn University, Bangkok, where he also serves as the head of the Center of Excellence on Natural Products for Neurodegeneration and Anti-Aging. A leading authority in neuropharmacology, Dr. Devin has dedicated his career to advancing the science of the healthy aging. With a prolific portfolio of over 200 publications, his work has garnered more than 10,600 citations and an H index of 36, marking a significant contribution to the global understanding of natural compounds. His research laboratory utilizes sophisticated experimental models, including cell culture, yeast, and animal model, to decode the molecular mechanism through which natural products combat neurotoxicity, neuroinflammation, and the complexity of neurodegenerative diseases. Dr. Devin will be speaking on the topic Integrated Model Approaches for Identifying Natural Products Against Neurodegeneration and Aging. We warmly invite Professor Dr. Devin to take the podium.
Thank you very much for the introduction. I also would like to thank the organizer for giving me this opportunity to share my research work, but I wouldn't talk too long. Okay. My background, I studied with the Virginia Commonwealth University in the US, and that time that I studied, I discovered the mechanism to control the gene regulation of the galactosylcelulosid, which is the spingle lipid in the myelin sheath made by the cell called oligodendrocyte. So that what introduced me to the field of neuroscience since then. And I published 2 papers that almost 25 years ago for my PhD dissertation. And after that, I still believe that the lipid may have something in the function or contribute to the function of mitochondria or the cell in the neuronal system as well. But lipid, if you look at them carefully, it could be a beneficial factor. But somehow in this paper that you can see here, it's not good that you have the lipid droplet and also interfere with the mitochondrial function. So since then, I think lipid may be very important to the brain, particularly because in the brain, particularly in the myelin sheath, it composed of the lipid 70%, whereas only 30% of the protein, which is contrary to other lipid protein composition in other cells. So this is very unique in the brain lipid and protein composition. About global aging, Thailand has very unique population. If you can look at this slide, as you can see, we have almost on 25% of the population with aging on already. In fact, 10 years ago, we are number 10, but we still top 10 with people with 18. But I think next 10 years from now, we may have double population 18. So that contributes to number 6 in the region, Asian region. And number 1 still be still Japan, number 1 of 18 people in the country. So that contributes to the global aging. And I want to show you this slide, very important. When we have global aging like this, there are some disease which are age-related disease, as you can see here. Cardiovascular, cancer, for example, type 2 diabetes and dementia and Alzheimer's disease. I will focus more on Alzheimer's disease. With population aging in the country, there will be very critical problem with 3H. Number 1, hypertension. Number 2, hyperglycemia, which is type 2 diabetic, for example, and hyperlipidemia, when you cannot control the fat metabolism in the body. That would lead to the cardiovascular disease. This is my university, Chulalongkorn University, located in the heart of Bangkok, very beautiful campus. And also, as you can see here, we have variety of natural products in the country, from the north to the south, to the west, the east. So the Thai government really want us to have some sort of plant extract or natural compounds, natural phytochemicals, something like that, to promote the health prevention system in the country. So with the grant related to natural product study, we can get a lot of money. And of course, with the collaboration with you guys sitting here, we can get even more, even better research funding. In the past, it's been very difficult, actually, more than 100 years already. Still, we don't have the way to cure the disease, particularly Alzheimer's disease. Still very difficult. Over 100 years, still, and I think in my lab, very small lab actually, we have more than 10 people, 5 postdocs, 5 graduate students. We want to contribute to the fighting. And when you talk about Alzheimer's disease, you can see these are hallmarks, some hallmarks like tau aggregation, beta amyloid aggregation, and also neuronal cell loss, something like that, inflammation as well. So when we do the experiment, we try to get the natural compounds, the plant extract, to fight against this aggregation or mechanism. So if we find any of them to work on this particular signaling pathway, and then we pay more attention to them. And as you know, mitochondria is very important. So I like to talk about mitochondria. And you can see the upper part of the photo. You can see normal mitochondria, and the lower part, Alzheimer's disease mitochondria. You can see large vacuole. So we found some natural compounds that we talk later that work to maintain the normal function of the mitochondria. However, we try to see whether or not it can reverse AD or Alzheimer's disease mitochondria to be the normal one. We still try to find that anyway. Look at the big animal like this. Would it be a good model for the lab? Like elephant. You can see elephant. So with this picture of the study, as you can see, it's very difficult to use any model and then translate to humans. They say that the mice not so good because it's too small. And then look at elephant like this if you can find some new information. Compare between human and elephants, you can see that only 5% of them die due to cancer, whereas for human, double die due to cancer. The good thing about the elephant is that they have 20 pairs of P53 gene, which is tumor suppressor gene. That may be a good way to escape from cancer, for example, whereas human only one P53 genes. However, the elephant weighs 100 times more than the humans. It seems that they are obese, maybe not so good because very heavy weight. But they can live very long, and also they die to cancer not a lot. Because of what? The conclusion is that because of they are vegetarians. So from this, you can say that eating or consuming vegetables, fruit, for example, would be really good thing for us. And then that actually published already. It's very good paper to study with regard to the comparison of humans and elephants. Look at this good fruit, vegetable, and also food, also like fish, for example. And with the vegetable, you can see many in the list like olive oil, avocado, red wine, vegetable, bean, whole grain, for example. So they are colorful fruits that mainly very good to the health. And in Thailand also, we have the boat like this. They have variety of fruits in there, and then some of them may be very good for us, but one of them I want to talk more today is the passion fruits. Based on the Nobel laureates, three of them actually, they just got award, Nobel Prize in economic science. They said that the sustainability could be happen because of our research can be actually really used in the future, that would be make it more sustainable. And also the very good thing is that innovation. So anything from knowledge, we have to try to get the innovation and then also translate to human use. The passion fruit is very good, and you can see the inner part with the white color. Only inner part with the white color has the activity. So we try everything. We try the seed, we try the fruit, and we try to compare with the inner part in the picture of the passion fruit. Only the inner part that had prominent activity. But I will show you here. There are some pharmaceutical activity known for passion fruit, like boost immune system, improve insulin sensitivity, but neuroprotective potential information is also very limited. So that's why we have to study even more in the lab. And then the question is that, is it good for health, good for Alzheimer's disease? Then that's simple research question that we have to take. So we extracted them, variety part, and also using different solvents to study them. And then compared to the hallmark of disease, we look at neuronal cell death, we look at mitochondria activity, we look at the oxidative stress in this picture, and then if the passion fruit extract that we study can really reduce this hallmark that we can observe. And then if we look at the picture of the cell, neuronal cell, you can see that there are dendrites, there are axons like this. So we can see even if some protein can translate from the end to the terminal of the cell, something like that. These are the models that we use in the lab. Cell lines, for example, neuronal cell lines. And then one of them very useful for us called C. elegans, which is the worm. This is very helpful for us to test the toxicity, to test the neuronal function, simple neuronal function, and we also use in silico analysis as well as the disease system. For Alzheimer's disease system, as you can see here, we use the model that called cell assay, like it could be cell-based assay, which when it challenged with the glutamate in a high level, the neuronal system, the reactive oxygen systems, we call ROS or reactive oxygen species could be double, for example. If any particular neuronal extract can reduce the reactive oxygen species or ROS, then we may do further research on that. So this is cell called hippocampal cell line, HT22, that we study in the lab. As you can see here, when the cell got incubated with the high content of the glutamate, the reactive oxygen species will be absolutely increased. And then we find a way to lower it somehow. And then here, as you can see, we use variety of techniques like confocal microscopic study, Western blot, try to really examine the natural products potential for the brain neuronal system, as you can see here. And then after using the cells for a while, I got contact by Professor Wink from Heidelberg University, Germany. He said, Germany, he said he has very good system, the model for worm, like C. elegans, could be used for my study. So we contact. So luckily, like Dr. Campbell said, we have Facebook and we have platform to really contact, and also very easy to communicate with each other online. That we got to really got contact and then start the lab C. elegans together almost 10 years ago. So the cell model to increase the reactive oxygen species like this, plus the C. elegans model would be combined, and then we can translate to the very helpful research data. I will show you some of them here also. This is the passion fruit extract that we studied. We found that the passion fruit extract can reduce the cell death. So when we incubate the cell with the glutamate, the cell will die. And then after, we also pre-incubated with the passion fruit extract and then glutamate, we can reduce number of cell death significantly. So saying that the passion fruit extract can really promote the cell survival. It could be negative, it can promote the cell survival, particularly in the neuronal cells, something like that. So this is very wonderful data. And then after that, we also check whether or not the passion fruit extract can really lower the reactive oxygen species inside the cell. So this is true answer. The reactive oxygen species can lower significantly when treated with the passion fruit extract, particularly the inner part of the passion fruit, as I mentioned, the white part of the passion fruit extract. Next, we study in the coelacanths. So move from cell model to coelacanth model. In coelacanth models, as you can see here, there are some glutaminergic neuron. For the Alzheimer's model, they have only a few neurons, but when we incubate with the passion fruit extract, we can remain the number of normal neurons to 6 or 7, not 3 after neurodegeneration like that. So we can track really the number of neurons, particularly glutaminergic neurons in the coelacanths. This is study in the coelacanths. And we also check the lifespan of the coelacanth. We found that normally the coelacanth will live like 20 days, for example, but when treated with the passion fruit extract, they can live longer. So saying that the longevity can be promoted in the presence of passion fruit extract. I will go fast for this. And also we can check the gene which is modulated by the passion fruit extract. So many genes, as you can see here. We also performed the transcriptomic as well, and we found that the gene modulated by the passion fruit extract mainly involved in the antioxidant gene like SOD, for example. So this is very nice experiment data that we show here. This is the conclusion in the part of passion fruit extract. Study both cells types and the coelacanth worm. And we had one student, she was a Chinese student who studied undergraduate degree in U of Malaya, and then she went to my lab, Chulalongkorn University, for her PhD. And then during her PhD thesis, before the COVID era, I sent her to Norway for my collaborative work. Since then, she never came back because of the COVID-19, but we got out a very good paper, but eventually she got PhD from Chulalongkorn University, and during that time, we made a very good collaboration with the Norwegian people as well. And we found that the two compounds you can see here, from the passion fruit extract, one called alpha amyrin, and the second one in the chemical structure here called beta amyrin. So the two of them, alpha and beta amyrin. And then we have to check which one really the main actor to fight against Alzheimer's disease phenotype. So we have to, maybe they can work together, or maybe only one of them work together. Then leading to the current research, and we just got publication 5 days ago. You can read online in Advanced Science. And this is the title that we published 5 days ago. After so many years of the study, we found that the mitochondrial guardian, only alpha amyrin, can mitigate Alzheimer's disease pathology via modulation of the DLK1 ULK axis. ULK gene actually can really work in the mitochondria by reducing the waste like toxic waste, or something like that. This is very important paper. But to this finding, to rule out beta amyrin and remain the alpha amyrin for future use, maybe it could be for commercialization in the future. Actually, this is the study that we really did in that paper, Advanced Science. We had people who consume the colorful fruits for 10 years, compared to the group that not really eat the colorful fruits. and we found very different in terms of the phytochemicals and also the change in the phenotypes. And also we actually use the AI also to predict the pharmacological network, which kind of drugs would be the potential ones. And we found that alpha-marine is very important. And then after that, we found in this experiment, we found that alpha-marine can reduce the tau aggregation, and also it can really promote the tau degradation. So this is very important. The tau is very important for Alzheimer's disease hallmark as a marker. And I think the idea from now, we may also check on the drug already in the market that may not be used for the Alzheimer's disease, but they use for something else. For example, this is anti-hypertension drug. When we test, somebody else actually, they tested with the coelacanth and found that the drug used in the market for the disease can also extend the lifespan in the coelacanth product. As you can see here, I think when we work with the current drug, it could be really launch to the market faster. Otherwise, it may take a long time to launch the market. This is my research group at Chulalongkorn University. You can also find us in the Facebook as well. This is my last slide. We will have the meeting in Thailand in May, after mid-May, about AI bioinformatics in the study of biology, aging, age-related disease. So if anybody would like to join us, we welcome you all. Thank you so much. I take it.
Thank you so much, sir, for giving a great presentation on your research. So I think Dr. Dinesh has also discussed about the phytomedicine, and you are also discussing present food. Sir, can you please explain what is present food and how to take? Because these are the diseases which you have mentioned, eczema, hypertension, hypertension. It is commonly found across the globe. So what are the precautionary actions can you take into this society?
About the present food, right? We actually, this is the species called P. edalis. So I think we have one species, and then we just got them extracted. We never thought it's going to be so far like this. Just simple, really simple PhD that time. In fact, I just want to really compare the passion fruit that grown in the north and the south of Thailand. Actually, just that's it. And then we found that somehow we used them with the brain cells, and we found it had very good activity. And then we searched the literature whether or not they're really good to be used. Actually, we found really basic phytochemicals in the passion fruit. And also somebody else already studied, some of them, published that passion fruit has a very good or very nice potential to the health. But nobody really talk about the neuroprotective potential. They talk about anti-diabetic, for example, anti-cancer in some of them. And I think we should really first go for better animal models for the study. And then after that, we see really two of them prominent compound that may be really good one, alpha and beta amyloid. And then we try to patent both of them, alpha and beta amyloid, but we could not do that successfully because the group of the people in Korean scientists, they try to patent it. They submit the patent in Korean society somehow, but they got rejected. Even they got rejected, could not be patented. They said it's not novel anymore. So what we done at that time? We have to modify the chemical structure of alpha beta amyloid. We got the grant from Norway, collaborative with the Norwegian group actually, and try to modify the phytochemical structure of both. Still, only alpha and beta amyloid still the best. And then we thought they could work together, but actually they're not. So we have the Alzheimer's model, mouse model in China, studied collaboration in China, and then we really got to this by only one of them that works very well. And further, we have to study more like what we see in the products or in terms of pharmacodynamics in the future.
Thank you so much, sir. Thank you so much.
Thank you so much, sir, for the insightful presentation. Now I would request Dr. Rahul Pratap Singh, sir, and Dr. Zafar Khan to kindly felicitate Dr. Tevin. As we have headed towards the end of the plenary session 1, I would request to acknowledge, I would like to acknowledge all the speakers with a round of applause for an enriching and thought-provoking session. And finally, I would like to request Dr. Rahul Pratap Singh, sir, and Dr. Sahil Arora to kindly felicitate Dr. Zafar Khan for chairing this session and for his valuable presence. Give a round of applause, everyone. Thank you so much, everyone. Now we will be breaking for lunch, so I would request all to please disperse towards the student refractory and the guests. And also I would like everyone to reassemble to the auditorium for the next technical session sharp by 2:45 p.m. Thank you, everyone.